Hi everyone, hola, welcome to my presentation about molecular and morphological species delimitation, such as the single species of the beetle-spider genus Ballus in Sri Lanka. I'm Dilini Bopearachchi. At a glance, there are three similar jumping spider species of the genus was recorded from Sri Lanka, so I'm using the molecular phylogeny to find out whether those species result single species or to find out the different species. I selected the family Salticidae of the spider world. They are the first and largest family of the spiders. It contains 661 genera, contains 6,379 species. The jumping spiders are very special ones because they are scrappy carnivores, they are ambush hunters, they have the characteristic eye arrangement, they can insect mimics and camouflage, and they can jump over 50 times of its body length. There are a lot of comprehensive studies on morphological, behavioral, and ecological and molecular aspects carried out about the jumping spiders in the world. And jumping spiders are member of the Araneae clade, and they are oval chelamitrin clade in the dinotia. And here you can see the position of the spider tree of life. It contains Salticidae. Initial work of the jumping spiders in Sri Lanka was started by the Eugene Simon, a French naturalist, in 1930, and according to the National Red List of Sri Lanka in 2012, it contains 97 species belonging to 51 genera were recorded from Sri Lanka jumping spiders. It has a large endemic component. Every year, jumping spiders numbers are increasing. The last review on phylogeny of the tribe Darwin was done by the Benjamin in 2004. They have the unusual various body forms in tribe Darwin species. Some are like the beetles, like the Ballus, and some of the like the ants, like Maringo. And the genus we know better, Ballus species, is recorded and identified by the Simon in 1990. The genus Ballus contains in the world 9 species, and they are distributed in the world. But no Ballus species were described from Sri Lanka after Benjamin in 2004. The genus Ballus is monotypic genus and it was endemic to Sri Lanka. Then Simon found 3 species from Sri Lanka, but out of 3, 2 valid species and another species, Ballus Koch, only he listed, he didn't describe that one. And those species are morphologically similar with the European Ballus. And Ballus segmentatus and Ballus sellatus that collected from the Simon is the type localities Agarapatana, and he collected Ballus sellatus collected Ballus sellatus type specimen only female and Ballus segmentatus only a male, but no drawings we cannot find. So find out whether these 3 species are single species or the different species and how many species were recorded Ballus in Sri Lanka. To find out, I carried out this research. The objective is research to assess the number and the distribution of the Ballus species in Sri Lanka. To sample the jumping spiders in Sri Lanka, I carried out the fieldwork in all climatic physiographic regions of Sri Lanka. And I collected the jumping spiders from the beating vegetation and hand collection, specimen preserving either 70% or 100% ethanol. And I did the taxonomic drawing with the aid of the Olympus BX51 compound microscope that's attached on with the drawing tube, and digital auto-mounted images from the Leica camera mounted on the Leica stereo microscope. For the molecular analysis, I selected as Neops taxa. I knew the sequence Ballus specimens for 17 specimens used, and additionally 5 Ballus species of the interest were obtained from GenBank. For the outcrop one, I selected 5 taxa belonging to 5 genera: Systematists, Lycum, Pedilla, Metistata, and Evergreen. And these are the gene targets and conditions that were successful for my research work. And the DNA extraction was done by using the conjugate DNA tissue kits, and genomic DNA was typically extracted from one or two legs, and polymerase chain reactions was carried out using the normal method, high multiplex PCR method, and also the PROTAC 3D method. Then all purified PCR products were central sequenced. And after central sequencing, sequence edit and assemble using the Genius 11.0.2 software, and the sequence alignment done separately per gene for the Muffet using high accuracy GINS setting. Multiple sequences alignment checked by an eye in IView. A phylogenetic tree based on the concatenated genes inferred using the IQ tree. And I employed three common use algorithms for the de novo species delimitation: tree-based multivariate question tree process, a distance-based clustering approach, and the BPP method. Then I would like to move to the results and discussion part, mainly contains the morphological delimitation and the phylogenetic and the species delimitation analysis. In morphological delimitation, the SIM found two species, Ballus segmentatus and Ballus sellatus from the and Ballus segmentatus specimen was only male, and the female found from the Ballus sellatus. There could be a doubt was arise in previous researchers. It could be sometimes maybe a different two species, or sometimes maybe a same species, male and female. So I used the 28 Ballus samples I collected from the localities to find out whether if there are special characters to differentiate those Ballus specimens, but no characters that could be used to unambiguously diagnose more than one species. Some observed variations was find out some color pattern and color pattern likewise, but it doesn't warrant the recognition of the two or more species. Then I carried out the phylogenetic and species delimitation analysis for these samples. This is the phylogeny of the Ballus based on the concatenated partition matrix of the CO1, 28S, and H3 genes introduced with the IQ tree. It contains 1,525 base pairs. And here you can see the monophyly of the Sri Lankan Ballus was recovered, and it was well supported. But however, with Ballus segmentatus nodes that are not related creatively supported, but together with you can see the internal branch lengths of the Ballus segmentatus clade, which results endorse the presence of the single species of Sri Lanka. And also this phylogeny recovers the special sister group relationship between the Ballus segmentatus and the Peperomythus, other than the Ballus chelibius from European and the Ballus segmentatus from Sri Lanka. And these delimitations suggest that Ballus be a paraphyletic. And here I taught all the molecular phylogenetic delimitation analysis I used, APGT, MPTP, and BPP, that confirms the presence that all the samples that conducted was confirms the presence of the single species of the Sri Lanka. And MPTP infer the zero support of speciation event in the all nodes of the Ballus segmentatus, except the one, the basal one, near the 0, 1, and also the that one MPTP also find out the only presence of the single species, and APGT also infer the exact single species presence of Sri Lanka in all initial intraspecific distances and all the initial and the recursive partitions, and it rejects the presence of the more than one species. And in the BPP analysis also, it also found some genetic structure among the Ballus in Sri Lanka, but it does not support to presence the more than one species. And I would like to go into the concluding remarks. This study find out the mystery of the Ballus results, and it proposed the single species of the Ballus is present in Sri Lanka. And the morphological delimitation, all the characters and the morphology are used. All the study sample justifies the synonymizing of the Ballus sellatus synonym 19 under the Ballus segmentatus synonym 19. And also the Ballus characters, it's Simon only mentioned that we can describe and we couldn't find out, so it's remained the normal notum. It's by the Bonnet in 1955. And using other than the morphological delimitation, the phylogenetic and molecular species delimitation, so we find out that it mentioned that presence of the one single species of the Ballus in Sri Lanka. So this study confirms there is a high potential of the molecular species delimitation as a useful tool because that complements the classical taxonomy and has a great potential for resolving taxonomic problems. Because normally in the spider world, we are using, traditionally we use in the primarily morphology to find out, differentiate the species and to find out the genus. So this molecular species delimitation is a useful tool for resolve the complementary classical taxonomy problems. So I would like to wind up my presentation. I would like to acknowledge National Institute of Fundamental Studies in Kandy, National Research Council Sri Lanka, Department of Wildlife and Conservation, Girls Department of Sri Lanka, Department of Archaeology, and the research assistants in evolution, ecology, and biodiversity research group, and also my family members. And these are some references I used for making this presentation. Thank you.